Hello everyone. Previously, we took aim at the video A Critique of Universal Common Ancestry from the channel Faith Because of Reason. Thanks again to Kyle Allender of the channel Christian Idealism for pointing us to this video. So, let's jump right in. I saw a comment on Facebook regarding part 1 that said the title of the video was misleading. For clarity, the title is a reference to the title of the video being critiqued. Since that one is A Critique of Universal Common Ancestry, and the author is an intelligent designer putting forth ID arguments, I figured, why not title it A Critique of Intelligent Design? Oh well. Let's get back to where we left off.
Perhaps you've seen a picture of a bat wing, a dolphin flipper, a horse's leg, and a human arm all side by side. The picture is intended to demonstrate that although the four appendages serve different purposes, they all have a common structure. These functional similarities are known as homologies. Darwinists point out that this common structure is easily explained by the theory of common ancestry. This is explained just as well by the theory of common design plan. Consider that there are many examples of where a common plan is used for different constructions by intelligent agents. Think of automobiles. Almost all automobiles have four wheels, an exhaust pipe, an engine, a steering wheel, and so on. These similarities are certainly not due to common ancestry. Indeed, evolutionary biologist Tim Berra tried to use automobiles as an example of descent with modification. However, this example backfired, since the cause of the change in these vehicles was intelligent design. Thus, this example actually served to undermine his point and so has become known as Berra's blunder. The fact of the matter is that intelligent beings use useful models and plans over and over again for different purposes. Thus, homologies are explained equally well by a common design plan as they are by common ancestry. According to Jonathan Wells, it is clear that there can be other explanations for a sequence of similarities besides descent with modification. In fact, most pre-Darwinian biologists would have explained such sequences by creation by design.
Similar structures are better explained by common ancestry than common design, especially similar structures serving different functions or different structures serving the same function. For example, why do whales have tetrapod limbs with the exact same bones? Humerus, ulna, radius, carpals, metacarpals, and phalanges. This is very different from the structures found in sharks. Different design, different designer? Why are bat wings supported by 5 fingers, and those of birds are supported by the fusion of 3, and those of the extinct pterosaurs by an elongated ring finger? Different design, different designer? Why are the eyes of mollusks and vertebrates very similar on the surface, yet fundamentally distinct? Why do all vertebrate eyes have the signature blind spot, which is not present in the eyes of any mollusk? Different design, different designer? The point is, it's not just similarities, it's the pattern of both similarities and differences among organisms that form a twin-nested hierarchy, which is evidence for common descent with modification. And the modified characters are dependent on the ancestral condition. Once a structure evolves, it can then be co-opted for new purposes as evolutionary pressures shape species. Hence why whales have tetrapod limbs, and modified into a flipper instead of borrowing the design from fish. This is also the reason we don't find chimeras like a Pegasus, which phylogeny cannot account for. Also, FYI, the platypus is a monotreme. This doesn't apply to products of design, like cars, where the designer isn't restricted by the rules of evolution and phylogeny. They are free to swap parts from different models, or they can redesign the whole thing and start from scratch. This is why you can't construct a cladogram based on their traits. Aaron Ra's video, Falsifying Phylogeny, explains this more thoroughly. Common descent necessarily predicts a nested hierarchy, while intelligent design doesn't. You can't just shoehorn design in wherever you want. Remember, the author never gave us a clear picture of how much he thinks is related to each other. In this video, he has expressed that some organisms are indeed related to each other. Therefore, in those cases, their structures would in fact be homologous. But how much homology does he allow? If the pectoral fins are homologous between lionfish and yellow tangs, Are their pectoral fins also homologous to the pectoral fins of cichlids, seahorses, eels, and other fish? Or were they created separately? And how would he be able to tell the difference? He can't. Think about the analogy he provides. Personally, I don't like analogies because creationists tend to abuse and reify them. I'll give a for instance. Creationists like to say that because DNA and computer code have some superficial similarities, DNA must be designed too. The problem is that DNA varies through natural processes, i.e. mutations and recombination, unlike computer code, and researchers understand how changes in DNA can give rise to adaptive novelties. Computer code and cars don't do anything even remotely similar. Additionally, there is a huge difference between how humans design things and how organisms are laid out. The hallmark of human design is simplicity, not complexity. Humans want to make things as simple as possible, but organisms bear the stamps of their ancient ancestry. That's why the human genome is only about 20% functional, why we have pseudogenes, endogenous retroviruses, and other selfish genetic elements, and why we have structures like the palmaris longus muscle that don't do anything. We've been cobbled together from 4 billion years of evolutionary history. As for the Jonathan Wells quote, duh, I guess. Of course, the pre-Darwinians would have invoked design, that's all they had. Nothing is especially shocking about that. Next.
A very popular argument for evolution is the claim that humans and chimps share 99% similarity in their DNA. The first thing to notice is that, at best, this only demonstrates that humans and chimps are related. This does nothing to demonstrate that all forms of life are related. However, it is not even true that humans and chimps have 99% DNA similarity. While loudly trumpeted by Darwinists as near-conclusive proof of evolution a few years back, more recent studies have revealed that human and chimp DNA is closer to 97% similar, and some estimates are as low as 95%. Of course, some may argue that these numbers are still comparatively close, but when one considers just how vast the amount of information in the genome is, even 1% difference becomes a substantial amount. Additionally, the 97 to 95% similarities that exist are confined to the so-called bodybuilding section of the genome. Since chimps and humans do share a somewhat similar body structure, it shouldn't be all that surprising that they share a degree of similarity when it comes to their bodybuilding DNA. It's not even clear that more similar DNA indicates closer ancestry. Consider that a common mouse has some genes that are also 99% similar to humans, yet humans are not considered to be very closely related to mice. Fazale Rana and Hugh Ross say, the most comprehensive genetic comparisons indicate that humans and chimpanzees share a genetic similarity closer to about 85% than to 99%. From an evolutionary perspective, if a 99% similarity reflects a close evolutionary connection, then an 85% genetic similarity distances humans from chimpanzees.
I hate this vague usage of the word information. It is so often a useless buzzword. I find it rather amusing that the author is okay with humans and chimps being 95 to 97% similar genetically, saying, quote, "At best, this only demonstrates that humans and chimps are related," close quote. I do have to agree with the author that even very small differences in the genome can contribute to relatively large morphological differences. The genetic similarities between unicellular and multicellular algae being a very good example on this point. Also, some mice genes are probably 99% similar to our own, but generally, for any given mouse gene, the chimp version will likely be more like our own. There's that nested hierarchy again. The jump from 99% to 96% is explained by the fact that at first we could only compare sequences with single nucleotide differences, or SNPs. Based on those, we are 99% identical. However, there are also large-scale changes such as duplications, insertions, deletions, and rearrangements of the chromosome. The fusion of chromosome 2, to name one specific example. It's difficult to compare such things in a simple way since it isn't simple. So now when we include those other changes, we are approximately 96% identical. Though important in phylogeny, the exact degree of similarity isn't specifically what demonstrates common ancestry between us. It's the shared genetic changes that must have happened in a common ancestor. A good example are ERVs and the nanog processed pseudogene family, which are unique insertions in the genome. If two organisms share the same insertions, that insertion must have happened in the genome of a common ancestor. Yet we share dozens of ERVs and the nanog pseudogenes with chimps. Creationists often jump on the fact that some ERVs are functional, but that's irrelevant, since the point is that they are unique insertions that could only have happened in one individual at some point. Thus, shared ERVs must have been inherited from a common ancestor. Creationists also like to point out apparent contradictions, such as chimps and gorillas sharing some ERVs which aren't present in humans, but this ignores simple population genetics, such as incomplete lineage sorting. And another common argument is that ERV insertion isn't entirely random. However, it is far from locus specific, such that it is still highly unlikely that the same ERV inserts itself in the same place of the genome of two different individuals independently. As for the Rana and Ross quote, that 85% similarity number is wrong. One possible origin for it is the creationist Jeffrey Tompkins, but then why are two old Earth creationists relying on a young Earther? Regardless, Glenn Williamson of the blog Ruhif has done a number of takedowns of Tompkins' material, so check those out for more information. Apparently there's no citation at all for this claim in Rana and Ross's book. Oh well. Next.
In his best-selling book The Language of God, Christian evolutionist Francis Collins argued that junk DNA provides powerful support for evolution. The vast majority of the human genome does not code for proteins, and so Collins assumed it was junk. Essentially, Collins argued that because most of the human genome is functionless, it makes no sense that God would create humans in their current form with so much useless DNA. But it makes sense if humans are a product of a long evolutionary history. The key assumption of Collins' argument is that there is in fact such a thing as junk DNA. However, thanks to the ENCODE project, we now know that at least 50% of our genome is functional, and perhaps as much as 80%. DNA does more than just code for proteins. It performs other crucial roles as well. With this research raising the numbers so drastically, it looks promising that functions will be discovered for the remainder of the genome. As Jonathan Wells says, given the abundant and growing evidence for the functionality in non-protein coding DNA, it seems that recent defenders of the myth of junk DNA are motivated by something other than scientific evidence.
And the ENCODE canard. What a shock. The purpose of ENCODE, aka the Encyclopedia of DNA Elements, was created to identify functional elements of DNA. Despite this potentially useful goal, the project became mired in controversy when it defined function so broadly such that a sequence was considered functional if it was transcribed or associated with a binding protein at least once in at least one cell type. Clearly, claiming everything that is transcribed as functional would lead to a lot of false positives. And as Dan Grauer points out, by such loose criteria, you could say that the entire genome is functional, since everything interacts with polymerase when the genome is duplicated. To be sure, genes do different things. Some code for proteins, some are regulatory. That doesn't mean everything which is transcribed does those jobs. Junk DNA is also not a synonym of non-coding DNA. Functional non-coding DNA, such as promoters and operators, were already known even when Susumu Ono published his paper, So Much Junk DNA in Our Genome, in 1972. And he didn't argue for junk DNA just because scientists don't know what the rest of the genome did. One major reason for why a large portion of our genome has to be non-functional is that the genetic load would otherwise be unbearable for our population. Ono predicted that the number of functional genetic loci couldn't be greater than 30,000. Bear in mind that humans have between 20 to 25,000 genes, so Ono's estimation is still quite accurate. More recently, Dan Grauer published a paper where he shows that 25% is the upper limit for the functional portion of the genome, while 10 to 15% is a more reasonable estimate. If it was higher than that, every couple must produce an unreasonably high amount of children on average just to compensate for the mutation load in order to maintain our population. And if junk DNA does provide important functions, there's also the question of what does it do? And why does it vary so widely between even closely related organisms? Why do some organisms, like onions, need so much, and some, such as fugu, need so little? This is at the heart of T. Ryan Gregory's onion test, which is a simple reality check for anyone who thinks they have come up with a universal function for non-coding DNA. Furthermore, the argument that the existence of junk DNA is necessary for Darwinism to be true is, ironically, completely backwards. Darwinian evolution in the strict sense is just mutation and natural selection, yet the accumulation of junk DNA is the product of random genetic drift. In other words, it's an example of non-Darwinian evolution. Refuting junk DNA wouldn't mean the end of evolution as a whole, far from it. It is purely a creationist myth that so-called junk DNA was always thought to be entirely functionless. Tony Reed did a whole video on junk DNA, so go check that out. Next.
Evolutionists such as Ken Miller are fond of using human chromosome 2 as evidence for evolution. The claim is that this chromosome is actually two chromosomes fused together. This would mean that at one time humans shared the same number of chromosomes as chimps, our supposed ancestors. It should be first pointed out that the evidence for this fusion event is less than convincing. Telomeric DNA at the ends of our chromosomes normally consists of thousands of repeats. However, human chromosome 2 contains much less telomeric DNA than we would expect if it really were two fused chromosomes. Human chromosome 2 contains only 158 repeats. Additionally, the telomeric repeats that do exist are highly degenerate, meaning that they have diverged a lot from their original state. This fusion event would have happened recently, and it's not likely that this much degeneration could have happened in so short a time. Lastly, telomeric DNA is somewhat common in mammalian chromosomes. Therefore, the telomeric DNA is not necessarily an indicator of a fusion event. Nonetheless, it is entirely possible that such a fusion event really did occur. But so what if it did? The event clearly happened after humans supposedly departed from their common ancestor. Thus, the fusion event itself tells us nothing, since it is equally compatible with a monophyletic view of life as it is with a polyphyletic view. But if this fusion really happened, that means we once had the same number of chromosomes as chimps. Doesn't this indicate common ancestry? Not at all. As noted earlier, we share a substantial amount of DNA with chimps, so pointing out one other genetic similarity doesn't imply common descent. Why is it so inconceivable that a designer might design humans and chimps independently with the same number of chromosomes? This is a perfectly logical and rational position to hold. Casey Luskin sums this up well. Human chromosome number 2 is not evidence for human-ape common ancestry. At most, it shows our lineage experienced a chromosomal fusion event, but it does not tell us whether our lineage leads back to a common ancestor with apes.
We've already done a video on human chromosome 2. Go watch that if you want to know the full story, but we'll summarize the relevant counterpoints. First, the argument that there is much less telomeric DNA at the fusion site than we would expect relies on the assumption that the chromosomes simply got stuck together at the tips, thereby preserving the entire length of the telomeric repeats of the ends of both chromosomes, which isn't the case. The fusion happened via a Robertsonian translocation, which always results in the loss of a large fraction of the original telomeric repeats, so no, completely backwards. The fact that the telomeric sequences in the fusion site is smaller compared to intact telomeric repeats at the ends of chromosomes is exactly what we would expect. Second, the argument that the telomeric repeats are too degenerate for a recent chromosomal fusion. Exactly what would count as too recent isn't specified, however, the fusion event could have happened as far back as just prior to the split between humans and chimps because of incomplete lineage sorting, about 6 million years ago. The fused chromosome could have been present but not fixed in the ancestral population. Then, after the split, the fused chromosome fixed in the line leading to humans and lost in the line leading to chimps. Third, the chance of finding one telomeric motif in the genome simply by random chance is 1 in 4.096. With 3 billion base pairs long, we would expect to find over 700,000 of these scattered throughout the genome and another 700,000 of the inverse complementary sequence. So, finding telomeric DNA in the rest of our genome isn't surprising, but that is just about one motif. What about 158 repeats? We don't find those throughout the genome, which isn't surprising. The chance of finding that by chance alone is 1 in 4.096 raised to the 158th power. The common calculator cannot handle such numbers. The exact probability contains 570 zeros. That's not considering the fact that telomeric motifs suddenly become the inverse complementary version, which is exactly what we would expect from a fusion event. And the odds don't get much better even if we take the degeneracy of the repeats into account. And fourth, the author has trouble understanding how this fusion could be evidence for common descent. After all, the designer could have made humans with the same chromosomes as other great apes independently of each other. But this completely misses the point of the predictive power of evolutionary theory as opposed to intelligent design. The latter has made no predictions concerning the number or nature of human chromosomes in relation to those of other apes. If chimps had approximately 100 chromosomes and humans had approximately 10 with no sign of any fusion or syntenic relationship between the chromosomes, the ad hoc answer from intelligent design would remain the same as is for all situations. The designer just designed it that way. On the other hand, evolutionary theory makes risky predictions that can be falsified, yet they are confirmed time and time again. Chromosome 2 is just one among many. Next.
Finally, we come to the vitamin C pseudogene argument for common ancestry. Most animals, though not all, are able to synthesize their own vitamin C by means of 4 enzymes. However, humans and some other primates have only 3 of the required enzymes. It appears as though we once had this ability, but that the gene broke and that we carry the pseudogene to the present. Thus, we cannot synthesize our own vitamin C, and so must obtain it through our diet. Ken Miller uses this as an argument for evolution. He writes, if the designer wanted us to be dependent on vitamin C, why didn't he just leave out the GLO gene from the plan for our genome? Why is its corpse still there? The common ancestor in which the capacity to make vitamin C was lost wasn't human, but primate, an ancestor that, according to advocates of intelligent design, we're not supposed to have. Although Miller is guilty of assuming that intelligent design requires one to abandon the theory of common ancestry, the basic point he's making is that since both chimps and humans have the same broken gene, it is likely that the gene broke in a primate common ancestor who then passed it on to modern humans. This is certainly a possible interpretation of the data, but it is not the only one. First, Miller's argument hinges on the premise that the vitamin C pseudogene is actually broken. It is true that the pseudogene doesn't code for protein. However, we know that not all genes do that. Many supposed pseudogenes actually are transcribed into RNA. We do not as yet know that the vitamin C pseudogene does this, but since the study of pseudogenes is still fairly new, it is far too soon to rule out that a function will be found. Thus far, studies give us reason to be hopeful, as many so-called pseudogenes have turned out not to be pseudogenes at all, but provide important functions. Second, it is entirely possible that this trait is shared between chimps and humans as a matter of sheer coincidence. Consider that guinea pigs also possess a vitamin C pseudogene that does not synthesize vitamin C. Guinea pigs are not thought to be very closely related to either humans or chimps, yet they also share this trait. Evolutionists conclude that this is a mere coincidence that the gene broke in the guinea pig. Fair enough. But if it can be a coincidence that guinea pigs share this in common with chimps, why can't it be a coincidence that humans do as well? John Sanford and Christopher Roop comment on this. It is not honest or consistent to infer that matching mutations in humans and primates proves common ancestry, while inferring matching mutations in humans and guinea pigs proves mutational hotspots. If the mutational bias argument is acceptable to explain matching mutations in humans and guinea pigs, why isn't it acceptable to explain matching mutations in humans and apes?
So, first, the author attempts to downplay how many other primates share the pseudogene called gulo with us. We share it, including the same mutations within the pseudogene, with tarsiers, Old and New World monkeys, and apes. All haplorhines have it, but no strepsirhines. Quite a coincidence, no? The author says, quote, many pseudogenes are transcribed into RNA, close quote, as if that indicates functionality. Genetic sequences that do absolutely nothing can be transcribed precisely because DNA is imperfect. It makes mistakes. Something doesn't have to be functional in any capacity to get transcribed. It just has to be in the path of RNA polymerase. It's called spurious transcription. We know the pseudogene doesn't work because we don't produce vitamin C. That's why we get scurvy when there's no vitamin C in our diet. And we already know gulo doesn't even function as an RNA gene since it isn't transcribed. It can't, since it's missing the promoter and all the regulatory elements. And even if it did produce a functional RNA, it wouldn't mean it's not a pseudogene in that it still lost its original function to produce a protein involved in vitamin C synthesis. Nor would that undermine it being evidence for common descent, since the shared mutations are still there. As for his argument that it's just a coincidence, this too is statistical nonsense. That would mean tarsiers, monkeys, apes, and us all broke the gene in the same way coincidentally. Intelligent design would have to explain why the designer chose to break the gulo gene in all haplorhines in the exact same way, independent of each other. The author mentions that guinea pigs have a gulo pseudogene too, and while this is true, it's broken in a different way than we haplorhines have. See the 1992 paper, Guinea pigs possess a highly mutated gene for L-gulanogamma-lactone oxidase, the key enzyme for L-ascorbic acid biosynthesis missing in this species. That's why it's considered a convergence. And based on sequence comparison, the estimated dates for the pseudogenization of this gene are very different between humans and guinea pigs, which are 61 and 14 million years respectively, which is just more evidence that their inactivating mutations are different and independent. John Sanford and Christopher Ruppy are then being either dishonest or ignorant, I'll let them pick, when they claim that the mutations in this pseudogene match in humans and guinea pigs. The mutations clearly aren't the same. Next.
Finally, consider that the human Y chromosome differs drastically in sequence and content from the chimp Y chromosome. Now, if similarity in the vitamin C pseudogene counts as evidence for common ancestry, shouldn't the drastic differences in the Y chromosome count as evidence against? It seems that for evolutionists, similarities count as evidence for universal common descent, but differences do not count against it. This has led Paul Nelson to ask, if species were not connected by common descent, how would we know it? Evolutionists apparently leave no room for the theory of common descent to be falsified. Evidence can only be offered in support of it, and contradictory evidence is conveniently explained away.
Ironically, even young Earth creationists like Todd Wood have taken apart the argument that the chimp Y chromosome is very different from the human one. Talk about a low bar. These creationists make the mistake of expecting that all chromosomes between humans and chimps should be roughly equally different from each other. However, there are several different reasons for why the Y chromosome experiences higher mutation rates compared to other chromosomes. First, the Y chromosome does not undergo homologous recombination during meiosis. Recombination is an important mutation correcting mechanism, ergo, the mutations that happen in the Y chromosome are less likely to be corrected. Second, since there is no recombination, any mutation that is under selection will effectively drag all the other mutations on the chromosome with it, even if they are neutral, resulting in selective sweeps. This is known as genetic hitchhiking or draft. Ergo, mutations on the Y chromosome are more rapidly fixed or lost from the population. Third, the Y chromosome is always passed on via sperm. The cells that produce sperm are constantly dividing throughout the lives of the males, whereas those of eggs are not. Ergo, higher mutation rates within the Y chromosome. And fourth, it's also true that males have a greater potential to pass on their genes, since they can theoretically have hundreds of children within the same time that it takes for a female to bear just one. Therefore, fixation rates of mutations on the Y chromosome in the population can be a lot higher. These and other factors explain why the differences between the human and chimp Y chromosome is significantly larger than those of other chromosomes. So it is not a problem for evolution. Creationists have a problem understanding basic genetic inheritance and population genetics. The author is also perhaps being a bit disingenuous in claiming that the differences don't reflect on evolutionary relatedness. The differences that are present in the chimp Y chromosome are certainly acknowledged, but they also must be factored into the whole of studies on human-chimp synteny. And let's answer Paul Nelson's question about how we could know if common descent were untrue. Well, for one thing, testable predictions that scientists make based on common descent wouldn't be constantly verified again and again. Or we can make observations that are incompatible with evolution, like the aforementioned Pegasus or Berlinski's example of a finch evolving into an elephant. Another really good way to know that we're not related to anything else would be if every Barrowman, or whatever you want to call each created group, had its own genetic material. If the genetic material, or developmental plan, or body plan of every Barrowman were completely different, then that would be terrific evidence of independent creation. There are virtually infinite ways for a creator to design life such that it would be crystal clear that they aren't all related. And it would certainly be no issue to create for an infinitely powerful being. Why don't we see that?
This point leads to another serious problem with the theory of universal common descent. Can the basic assumption of the whole enterprise, that more similarity equals closer relationship, even be trusted? Consider so-called convergent evolution, wherein organisms evolve features nearly identical to other organisms independently. Whereas humans and octopi have eyes that are remarkably different from their supposed common ancestors, their eyes are nearly identical to each other. So if unrelated organisms can evolve similar structures independently, this casts doubt on the idea that similarity is an indicator of closer relationship. And if we can't reliably determine how closely related different organisms are to each other, then perhaps we should begin questioning the very assumption that all organisms are related in the first place.
First, the reason researchers can determine that convergent evolution exists is that the structures in question have fundamental differences that separate them. For example, the eyes of humans and octopuses, while both are called camera eyes, differ radically in their construction. So they resemble each other superficially but are nonetheless distinct. In humans and other vertebrates, light has to pass over the optic nerve to get to the photoreceptors. Cephalopods, though, have an eye constructed oppositely, which is why they don't have a blind spot while we do. No researcher comparing human and cephalopod eyes would ever confuse them. So this is still consistent with common descent. And as pointed out earlier, how does intelligent design explain things like this? Different design, different designer? Even still, it's not just the morphological similarities that determine relatedness, though those can contribute, but the genetic sequences. Genes position us among primates, while octopuses are among the cephalopods. Convergent evolution can sometimes obscure relatedness, but it doesn't generally, which is how researchers are able to identify said convergence. So, in totality, this video really didn't make its case. It threw together a bunch of intelligent design and creationist canards, most of which have been debunked 1,000 times. I highly recommend the author actually read the evolutionary literature, rather than merely repeat ideas. So, thanks for watching, and I'll see you next time.